So should we start?
Good morning.
Okay, my dear friends, very good morning. So, Rameshana, good morning. Hearty welcome. Your voice is not audible, Rameshana. No, unmute చేసుకోండి. Unmute చేసుకోండి.
Yeah.
Right. So, very good morning, my dear friends. We are going to listen about the new topic in our CPGET-2023 PG online coaching, free coaching. And also it is useful for all competitive exams in plant sciences like JL, DL, Group DL, JL, and PGT also. So, Madam today is going to explain about the Pteridophyta, general characteristic features, and the type studies of the Pteridophyta also. Today's our eminent resource person, Dr.A.Kavitha Madam Garu, Assistant Professor, Department of Botany, Rbvrr Women's College Autonomous, Hyderabad. Madam, please welcome and share your screen.
Okay, sir. Thank you so much.
Right. Welcome, Madam.
Yeah. Good morning to all the participants over here.
Morning, madam.
Yeah, good morning. So today onwards, we'll start with the Pteridophyta. Now, today I'm focusing only on the general characters, which are common to all the genera which are present in the pteridophytes of our syllabus. Just have my screen. I hope the screen is visible to everyone.
Yes, madam. Yes.
Yeah.
Yes, ma'am.
Okay, fine. Right. So now we are moving on to the next group of plants in the plant kingdom, that is the pteridophytes. So to starting with the pteridophytes, they are commonly referred to as ferns. We call them as fern plants. What is the meaning of fern? F-E-R-N. What is the meaning of fern? Fern refers to the leaf character. So the leaves of this plant, of the pteridophytes, they are appearing like a feather of a bird. Okay. So ferns are always referred to as feather-like leaf. The plants in the pteridophytes, the peculiar characters are the leaf characters of the plant body. Now, why we are focusing only on the leaf, I'll tell you in the coming slides. Now, so Pteridophyta, pteron, it is derived from two words. Pteron means feather-like and phyton is a plant. So pteridophytes are having feather-like leaves of the plant. Now, next, these are the first vascular cryptogams. Just like bryophyte, we call them as amphibians of the plant kingdom. In the same manner, pteridophytes are referred to as the first vascular cryptogams. What does it mean? That means to say, these are the plants which are going to develop the vascular system. That is the xylem and the phloem. The vascular tissues are going to be observed in the plant body. So these are the first vascular cryptogams. Now, so this pteridophytes, it includes both primitive and the advanced forms of living and the fossils. Now, these are all the genera of the pteridophytes. They are represented by 400 number and 10,500 species all over the world. And among them, majority of them are living and also some are fossils also. Now, another important point here is the dominant phase or the plant body of pteridophyte is designated as a sporophyte. I repeat, Pteridophyta plant body, the dominant phase of this pteridophyte is regarded as sporophyte. Whereas in bryophyte, the dominant phase was gametophyte. Okay. But here the plant body is considered as the sporophyte and it is differentiated into stem, root and the leaves. Whereas in bryophytes, we are not mentioning it as the stem, root, and leaf. We are not calling them as such because they are the smaller group of cryptogams. But here we can see the evolution of development of vascular tissues, and even the plant is also being modified and it has been differentiated into stem, root, and the leaves. So the plant here is represented as a sporophyte because the plant body will develop the spores on them. Hence, the plant body is represented as a sporophyte. And this is nutritionally independent of gametophyte. So in bryophytolo, we studied about the sporophyte depending on the gametophyte. But in the pteridophytes, it is completely independent. It is not at all dependent. Gametophyte is separate and the sporophyte is separate. And the adult plant body is sporophyte only. Now, so these sporophytes, the shape, size, and the habitat, they vary from one plant to the another plant. So in the pteridophyte, there are some small annual plants also, like Zola and Salvinia, and the larger plants like Angiopteris and the Marsilia, all that they comes under the larger trees. And most of these pteridophytes are the terrestrial plants. They always grow on the land surface. Now, so there are some aquatic species of these pteridophytes also. This is Azolla, which is aquatic, Marsilia, Salvinia, Isoitis. They all are the aquatic species. They are growing in the water bodies. When we go with the another plant, which is Selaginella rupestris, this is acting like an which is growing in the dry condition. It is a dry condition here, Selaginella lepidophylla, whereas it is a xerophyte. It can withstand the higher climatic conditions, or it can survive even without water also for longer period of time. So in the dry condition, this plant body appears like this. So when we dissolve that, this Selaginella in the water, it becomes the fresh enough. So this is a peculiar characteristic feature of this Pteridophyta, that is Selaginella lepidophylla. So this is called as resurcation plant. That is transformation of this, what we call it as the structure, or transformation of its nature from dry to wet condition. Next, we can also see the presence of the roots and the stem with a permanent growing apices, and some of them are herbaceous also. When we go with the leaves are sessile as well as the scaly leaves also been seen. In Lycopodium and in the Selaginella, we can see this is the Selaginella, this is the equisetum, this is the scale leaf, position of the scale leaf. And here this is in the Lycopodium, the leaves are sessile, they does not have any petiole. And this is also an adiantum, where we can see the presence of a small petiole. So the leaves of the Pteridophyta, either they can be the sessile or smaller in case of Lycopodium and Selaginella, whereas it may have a large petiole in case of this adiantum and the larger fern plants. And one more important point here is, in the larger plants of the pteridophytes, the leaf, the plant is identified by the leaf. Okay, this the Pteridophyta plant, the characteristic feature of identifying the Pteridophyta plant is with the help of the leaf. So leaves plays an important role, and they are also a characteristic feature of showing the circinate vernation. Now, what is the circinate vernation here? Young leaves. So that is the leaves which are not yet mature. They are under development. These leaves, they are coiled inwards like this. Okay, so in order to avoid any damage, or in order to protect it from the external very heat waves, they will be coiled inward like this. And one more important point is, these leaves, they have the growing tips, growing apices. So in order to protect that also, they are circinately vernated. So this is called as circinate vernation, coiling of the leaves inward. So this is called as circinate venation. Next. So depending upon the leaf structure here, the pteridophytes are classified as microphyllous and macrophyllous. Now, what are these microphyllous pteridophytes? So these are the group of the pteridophytes in which the leaves are sessile and the leaves are very simple leaves with a single vein, and the leaves are very closely attached with one another without any gap between them. You can look into this picture here. This is a lycopodium. It is having the microphyllous leaves. The leaves are very, very small. They are sessile and they are arranged very close to each other without any gap in between them. So these are called as microphyllous pteridophytes. Now, macrophyllous. So the name itself implies. Phyllis means the leaf. Macro, large size of the leaf. Okay. So the leaves are very large. They are pinnatified, that is pinnately compound like leaves. They are having a series of the veins in them, and they have a prominent gap between one leaf and another leaf. Okay, so this group of pteridophytes, they comes under macrophyllous. Next, we're moving on to the stem structure. The stem is usually branched in the pteridophytes. It can be a monopodial or dichotomously branched leaves, branched stems. And these branches, they do not arise from the axil of the leaves. The leaves and the stem, they possess trichomes, the hair-like structures. The stomata are present on both the side of the leaf, and the root and the stem, they are well developed with vascular system, which is composed of xylem and the phloem. And here there is, though the vascular, that means to say, though the vascular tissues are present, they are the primitive form. That is, they are the first developing vascular tissues. We can't see any secondary growth in them. Okay, that is, cambium is absent. They are only the primary vascular tissues. Okay, hence secondary thickening is absent. And some pteridophytes, they show some secondary growth like in isoetes, that is in the higher forms. Vascular elements, just not xylem and phloem, they are well developed. And presence of this vascular structure, that is xylem and the phloem, the vascular tissue, the steel, okay, the steel can be either of protostelic condition, siphonostele, or dictyostele or polystelic condition in the pteridophytes. So whereas the protostele here, where the xylem and the phloem separately arranged on two different strands, and they may be a primitive represented by only the smaller trachea of the xylem or the phloem parenchyma, like that. Only the simpler tissues are present. Siphonostelic condition, they are in the form of the tubular connections, and polystele, presence of many stellar strands. Now, xylem here is made up of tracheids. Phloem consists of seed cells and the phloem parenchyma. Whereas xylem, it is making the role of xylem, conduct the water and the minerals. Whereas the phloem is, the role of phloem is to conduct the food material. And the photosynthetic tissue here is restricted only to the leaves. Okay, whereas in bryophytes, we see the entire thallus or the upper surface of the thallus was preparing the food material due to the absence of the leaves. But when we come to this evolution of pteridophytes here, when we come to the pteridophytes, the photosynthetic region is restricted only to the leaves. Leaves are the one which are preparing the food material. And so there is no tissue differentiation in the microphyllous leaves. That is, the leaf is not differentiated into the palisade or the spongy tissue in the case of microphyllous. Whereas in the megaphyllous, that is a microphyllous, the leaves which are very larger run, there the leaves are differentiated into palisade and the spongy tissue. Now, when we go for the reproduction study, as I told you that the dominant phase of the pteridophytic plant is a sporophyte. The spores are the structures which are formed or present inside the sporangium, which are developed on the plant body. Hence, the plant is called as sporophyte. So the pteridophytes, majorly, they are going to reproduce by the spores. These spores, they are developed in the specialized structures called as sporangia. And these sporangia, they are born on the ventral side of the leaf. Okay, so that's why leaves plays a very important role here in the pteridophytic plants. So the leaves are the one which are going to occupy, which are going to give the space for the development of the sporangia. Now, the leaves which are going to bear this sporangia, we call them as sporophylls. Now, so in the aquatic form, this is regarding the terrestrial, what we told you. Whereas in aquatic forms like Salvinia or Marsilia, these sporangia, they are born in the specialized structures, what we call them as the sporocarps. So that we'll study in the type studies. Now, so the plants here, depending upon the development of the spores, the plants can either be termed as homosporous plants or heterosporous plants. Lycopodium and cherries, the examples where the spores are homosporous type, that is, the sporangia are producing only one type of spores. It can be the smaller spores or it can be the bigger spores. We call them as microspores and the megaspores. Whereas in case of Selaginella, the plant is represented or it is defined as heterosporous type. That means to say, in the sporangia, two different types of spores are developed, the microspores and the megaspores. Hence, the plant is referred to as the heterosporous type. Now, so this is regarding the homosporous. Just now I told you, development of only a single type of spores in the sporangia. And that sporangia, either it can be a microsporangia or it can be a megasporangia. So why we are calling it as a sporangia as microsporangia? They are developing small size of the spores, microspores. We call that sporangia as microsporangium type. And megaspores developed in the sporangia, we call it as megasporangium. And when we talk about the size of the spores here, the spores are peculiarly, they are smaller in size. Now, when I type of about the heterosporous type of sporangia, the sporangia which are developing the larger spores, okay, we call them as megaspores. And that sporangia, what we are going to, the sporangia which is developing that larger spore, that is called as megasporangium. And the smaller spores which are developed inside the sporangia, that is represented as the microsporangia, and the spores are represented as a microspores. But generally, oftenly here, we can say that in the pteridophytes, the larger spore is represented by a female megaspore and a female megasporangium. And the smaller spores are called as the male microspores, which are developed in the So it depends upon, again here, one more point regarding the sporangium here. The development of the sporangium is represented as eusporangiate type and the leptosporangiate type. Now, what is this eusporangiate type? If the sporangia, the microsporangia or the megasporangia, if the sporangia is developed from a group of initials, cells, we call them as sporangial initial cells. Development. This is again a very important point in the competitive exam. Eusporangiate, the sporangia which are developed from a group of initials. That development, we call it as eusporangiate type. And if the sporangia, if it is developed from only a single initial cell, then it is represented as leptosporangiate type. Okay, so this point to be noted, all of you. In the time of type studies, that plant, is it a eusporangiate development or leptosporangiate? We'll discuss at its sporangial development. Now, next we move on to the, after the sporangial development, these sporangia, where they are developed. These sporangia are always developed on the fertile leaves, which are called as sporophylls. I repeat this once again. The leaves which are developing the sporangia, those leaves are called as sporophylls. Okay, right. These sporophylls in pteridophytes, all these sporophylls which are having the sporangia, they are aggregated, they are compactly closed. They are, all these sporophylls, they are tightly or closely or compactly arranged with one another and forming a common structure, what we call it as a cone, or we call it as a strobili. Okay. I repeat, the dominant phase of the pteridophyte, it is a sporophyte. Why we are calling it as a sporophyte? Because they are developing the spores. Where are these spores are developed? Within the sporangia. Where are these sporangia are developed? These sporangia are developed in the sporangia. These sporangia are developed on the leaves, on the ventral side of the leaf, or in the axils of the leaf. And all these sporophylls, they are aggregated together, they are compactly arranged together and forming a specialized structure, what we call it as the strobilus or the cones. Okay, so this is a peculiar and the specialized character of the pteridophytes. These pteridophytes, they always develop the cones or the strobilus at the end of the stem, at the terminal part of the stem, in case of Selaginella, Lycopodium, or Equisetum. So this is the Equisetum cone, we call it as. This is a Selaginella. Okay, right. Now, so in the true ferns, that is in the primitive forms of the ferns, these sporangia, just now I told you, they are, so you can see these are the sporangia. Okay, they are developed on the lower surface of the leaf in clusters, and we call them as the sori. Okay, this is a sori, a group of sporangia which are developed in clusters at the ventral side or the lower surface of the leaf. So this one, this is the sori. You can see this is the sori. At the time of reproduction, they are developed. Now, so when we talk about the spore structure, so spore, again, it is having a very thick wall, often differentiated into outer exine and inner intine. These spores, they are germinating and forming the next generation, that is a gametophytic generation. Now, the homosporous species, that is the spores which are developing into, which are developing and producing only a single type of spores. The development of the gametophyte, we call it as hexosporic type of development. That is, the gametophyte will develop outside the spore wall. Whereas in the heterosporous, the gametophyte will develop within the spore wall, that is inside the spore. And here in the pteridophytic group, gametophyte and sporophyte, these are the two independent and separate plants. Now what is happening here? The spore, after liberated, that is after the spore, they are released from the sporangia or from the strobilus. When they fall on the suitable substratum, the spore will undergo germination and producing another generation called as gametophyte. And the younger gametophyte, you can look into this. This is a younger gametophyte of the pteridophytic plant, which is a thallus-shaped plant body. A thallid. The plant here, we can't see any differentiation of stem, root, and nothing like that. So it is a younger gametophyte, first formed gametophyte from the spore. So this is called as prothallus. This is called as a prothallus. That is the the thallus which is developed from the spore. Okay, or which can call it as a young gametophyte. So this prothallus, it is very simple, green and heart-shaped. So whereas in the, and also what is the role of this? Why we are calling it as a gametophyte? Why this it is green? So just now I told you, both are independent. Gametophyte and sporophyte, they both are independent. So they prepare its own food material. Why we are calling this as the gametophyte? Because it is going to develop antheridia and the archegonia, the sexual organs. So hence this thallus is representing as the prothallus or the gametophyte. Now, the homosporous prothallus, I'm talking about the homosporous prothallus. The prothallus which is developed from the homospores. That prothallus is a monoecious. On this gametophyte, antheridia and the archegonia, they are developed on the same one. Okay, so that's why it is a monoecious prothallus. Whereas heterosporous, just now I told you, the microspore, it will form the male prothallus, and the megaspore, it will give rise to the female prothallus. So because two spores and two sporangia and the two cones, they are different from one another. That's why it is the gametophyte which is developed from the heterosporous type of spores, they are always dioecious. And when we talk about the sexual organs, these sexual organs which are developed on this prothallus, they can be embedded inside the prothallus or they can be projected out of the prothallus. So here, so they are developed, they may be inside this gametophytic tissue or the sexual organs can be projected. And here the main sexual organ, it is represented by antheridium, which is going to be surrounded here also. It is surrounded by a sterile jacket layer of cells, and it is represented by only a single layer. And this antheridia, they are sessile or sometimes they are shortly stalked. And they are going to develop the antherozoids, which are of unicellular, spirally coiled, and with two apical flagella. Now, when we move on to the female sexual organ, the archegonia, it is also a same structure, just like what we observed in the bryophyte, having a globose venter and the neck region. And the neck, it is projected and venter is embedded in the prothallus. So the neck canal cells, upon the degeneration, it will develop the mucilaginous substance. So through which the antherozoids are attracted and they move towards the archegonium and fertilization is occurring. The egg and the antherozoid, they fuse to form the zygote, and the zygote will develop into the next generation, that is the adult plant. And here in the pteridophytes, the zygote is dividing and form the embryo. And the embryo, here we call it as of a proembryo. The first division of the zygote, it is determined by the polarity of the embryo, that is embryo upon the differentiation, it is going to develop the roots and the shoot apex. So here in the pteridophytes, they show a typical heteromorphic alternation of generations, having a distinct sporophytic and gametophyte, and the main plant body is called as the sporophyte, and it is considered to be as the dominant phase. And both them, they are independent. The gametophyte and the sporophyte, they are independent of each other. So this is a life cycle of a general life cycle of the pteridophytic plants. The adult plant here, I told you, the dominant phase or the adult plant is a sporophyte. Sporophyte is developing the spores. They are on the leaves. So here we are talking about the heterosporous type of life cycle I'm showing you here. This is a microsporophyll, megasporophyll. Microsporophyll will develop microsporangia. Megasporophyll develop megasporangia. Upon meiosis here, microsporangia will develop microspores, and megasporangium will develop megaspore. The microspore will form the male gametophyte or the male prothallus, and the megaspore will develop the female gametophyte. The male gametophyte often possesses the male sexual organ antheridium, and the female gametophyte will accommodate the female sexual organ called as archegonium. Antheridium will develop the male gametes, anthrozoids. Archegonium will develop the female gamete, egg. Fertilization is occurring here. Fertilization will result in the zygote. Zygote will undergo development and produce the embryo. Embryos are again, upon differentiation, it is going to form an end sporophyte and develops into an adult sporophytic plant again. So this is a typical life cycle of pteridophytic plant. So this is about the general characters of the entire pteridophytic plants. So I'm just stop sharing here. Any query here? Any question regarding the general characters of the pteridophytes here? From the participant side, please, if you want any point to be noted. So this is all the general characters which is applicable to all the genera of the pteridophytic plants.
Okay, my dear friends, any doubts regarding of this class? So madam is asking. Okay, otherwise I will wind up the session. Thank you very much, madam. Okay, madam, thank you.